welcome to BioXplaining. So this is the third part of our revision series for ICSE Class 10 diagram-based questions. And as the board exam is nearing, I know that all of you are feeling a little more anxious as to how will you all do your preparation all the more well. So yes, here I am with yet another part of the revision series. Stay tuned till the end of the video and practice well. Okay, so this is the first diagram that we have, and it shows us the structure of the chloroplast, right? Structure of the chloroplast. Okay, so let us see what are the different parts that are present. Number one is pointing towards the thylakoids, the flattened sacs, right? So you can imagine these thylakoids to be, say we can consider them to be a coin each. Okay. Number two is referring to a stack of those thylakoids, a pile of those thylakoids. So we call them to be the grana. Fine, grana and pile of thylakoids, right? So we can say that the grana basically looks like a pile of coins stacked on top of each other. Okay. Part number 3 is pointing out to the fret, which are the interconnecting things which join one grana to the other grana. Okay. Part number 4 is showing the grana in vertical section. Matlab say imagine this is a coin all stacked one on top of each other. You have made a section, you've cut it. Now if you look into it, this is how it is gonna look like. And part number 5 that you have is the stroma, which is basically the ground matrix or the ground fluid that we have. Okay, so the structure of the chloroplast is absolutely clear to all of you, I guess. Okay, so this is an experimental setup. The leaf shown below is put through the iodine test, and we all know that iodine test is used to identify the presence of starch, right? So we want to identify whether starch is present or not. Before the experiment, you have the central part is the green portion, outside part is the white portion. So what's going to happen to part A? That is your white portion. The white portion turns brownish, which shows that starch is absent. If you apply iodine, the white portion will either show not much change in color or it will be brownish. So that shows that starch is absent. What happens to the green portion is that this green portion shown by B here, that one, is going to turn blue-black, right? And that indicates that starch is present in this green portion. So the simple funda is that if you have your chlorophyll, that is the green portion matlab chlorophyll is present, right? If you have your chlorophyll, as simple as that, photosynthesis is going to occur. And we all know that the product of photosynthesis is glucose. Glucose will be produced. Glucose is stored as starch in the plant. So if you find on iodine test the portion turning blue-black, you can easily say that starch is present. That means chlorophyll is present and photosynthesis has occurred. Okay. Now the question is, why does starch gives a blue-black color with iodine? The reason is that the blue-black color is primarily due to amylose, which is a component of starch. So, when you all grow up again, those of you all will be taking up science and biology, in class 11 you are going to study that starch has two major components. One component is known as amylose, the other component is known as amylopectin. Okay, so this component, that is amylose component of starch, has a helical structure, right? So in what is a helical structure? The DNA-like structure, the helix. Okay, so this amylose component allows the iodine molecules to fit inside the grooves of this helix, to fit inside the helix. So as a result of that, it gives you a blue-black coloration. Okay, and in the absence of starch, you do not have any such component, amylose or helical structure. So the iodine doesn't know where to stick inside. So hence it doesn't give you a blue-black color. Okay, तो experimental result आपको पता था. Why is it like this? I guess that's clear now. Okay, so a question from the circulatory system. Blood components. Identify the type of blood cells and state major functions of each. Number A, part A, shows your RBCs. What are RBCs? These are the red blood cells, also known as erythrocytes. What's their job? They transport oxygen to body cells, and they do that in the form of oxyhemoglobin. Okay, so basically what are we trying to say is that your oxygen binds to the respiratory pigment, that is hemoglobin, and it forms an unstable compound, that's oxyhemoglobin. Okay, now this oxyhemoglobin will go to your body cells and dissociate into oxygen and hemoglobin. Now this oxygen will be used up by the blood cells to perform aerobic respiration, by the different body cells, I mean. Okay, then what's B? B is your neutrophils. So, one thing, carefully note, B, C, D, E, F, all 5 are your WBCs, leukocytes, white blood cells. B, C, D, look into their cytoplasm. What do you find? You find small granules. So you call these to be what? They are your granulocytes. Granulocytes. Okay. B also, C also, D also. All of these are granulocytes. And E, F, these do not have those granules. So you call them to be agranulocytes. Okay. So B, neutrophils, what is their job? They engulf bacteria. Fine. This is called phagocytosis. I hope we all know. So phagocytosis is basically, in simple words, we can say that phagocytosis is eating of the cell. Eating of the cell. Right. Next, C is your eosinophils. You can also give them another term. They are known as acidophils. Okay, what is the job of these acidophils or eosinophils? They engulf bacteria, secrete antitoxins, and they also have another job, that is associated with allergic reactions, associated with allergy. Okay, so these are the 3 jobs of eosinophil. Coming to the last, granulocyte. What is that? Basophils. Look into the names. One was neutrophil, neutral, eosinophil, acidic, basophil, basic. They are all stained by neutral, acidic and basic dyes. Like, neutrophils stained by neutral dyes, eosinophil by acidic dye, basophils by basic dye. What is the job of basophils? They release chemicals known as histamine for inflammation, which dilate the blood vessels. So this is important. You have to remember that they release the chemical which is known as histamine. Fine. Now E and F, there are no granules in the cytoplasm, so they are agranulocytes. E is your lymphocyte. What is its job? Its job is to produce antibodies. Lymphocytes can be of 2 types. One is your T lymphocyte. Why this name T lymphocyte? Because it matures in the thymus. Where does it mature? In the thymus. And another one is your B lymphocyte. What does the B lymphocyte do? The B lymphocyte matures in the bone marrow. Okay. The B lymphocyte matures in the bone marrow. Next, F is your monocytes. What does the monocyte do? Its job is to ingest the foreign substances. Okay. And at the site of infection, the monocytes will be converted into the macrophages. Okay, next, figure 24. The diagram shows a blood smear, right? Label the parts 1 to 4. 1 refers to the RBCs, right? So you can call the RBCs to be erythrocytes. 2 refers to your WBC. WBC also known as leukocytes. Part number 3, the platelets, also known as the thrombocytes. And what's 4 showing? That is the matrix. So that is your plasma, the liquid part of blood. Okay, so part number 1, 2, 3, all these 3 are your cellular component, the cellular part, and part 4 is the liquid part of blood. Clear till now? Structural differences between 1 and 2. Let's have a look. So your RBC is part number 1 and your WBC is the part number 2. Let's differentiate between the two. First point we can take is that RBC, they are disc shaped or biconcave, right? Talking about the WBCs, they are amoeboid in shape, right? Amoeboid in shape matlab that these are irregular in shape. Okay, means these are irregular. Some have like 2-3 lobed nucleus, some will have one single large nucleus, so there's no fixed shape for them. And another difference you can take is that for RBC, it lacks nucleus in the mature form. Mature RBC lacks nucleus, while WBC contains nucleus even in the mature stage. Okay, now the functions of 1, 2 and 3. So part number 1, its function is to transport oxygen to different body parts. Part 2, defense mechanism and immunity. See, RBC ka function we have just now seen, right? And then you have your WBC ka function, that is it will fight with infections, foreign substances, produce antibodies and all that. So defense mechanism and provide immunity to the body. Part 4, soluble protein found in part 4. Okay, that is 4 is the plasma, the soluble protein which helps in blood clotting. That is your fibrinogen. Okay, I missed out part 3. So part number 3 is the platelet, right? Plus 3 platelet helps in the clotting of blood, right? At the site of injury, the platelets will help in the clotting of blood. Fine. So I guess this one is clear. Okay, identify the diagram and label the numbered parts. So the diagram basically shows you that the pathway of entry of water from the soil, right, into the roots. That is how water particles are entering into the root cells rather. So part 1, very simple, that is your root cell where the water is entering. Part 2, the outer cells. So this is your epidermis or epidermal cells, whatever you say. Part number 3, the cells next. So that is your cortex or the cortical cells. Then on top, part number 4. What's that? That's your endodermis. So outer ones are your epidermis, inner ones are your endodermis. Then right before the endodermis or inner to the endodermis, part 5 is your pericycle, right? And then further inner to that, part 6 is your phloem. Okay. And finally, part 7. See where the water has actually now entered into. You know very well which tissue is responsible for the conduction of water and minerals upwards to the stem. That is your xylem. Right? I guess this one is simple and clear. A question from the excretory system. So identify the diagram. The diagram shows a kidney in longitudinal section. That means you have cut the kidney this way, right? Part number 1 refers to the cortex. That is the outer portion of the kidney. Okay. Part number 2 is referring to the inner portion. So that's known as the medulla. Fine. So cortex is the outer portion, right? And your medulla is the inner portion. Then part number 3 we have, see where it's pointing to, that's your papilla. Then part number 4 is pointing to this notch here, so that is known as the hilum. Part number 5, this expanded part in front, that refers to the pelvis. Okay. Then part number 6, these structures, these ones, these are known as the pyramids and their presence inside the medulla, so we call them to be medullary pyramids. Fine. And part number 7, the part which is taking the urine away from the kidney, that's your ureter. Okay? So I guess this one is clear to all of us. Chalo, identify the diagram and label the numbered parts. Extremely simple. This is the diagram of a singular nephron. Okay, so a nephron or a renal tubule, also known as uriniferous tubule. So basically a nephron or a renal tubule, this is known to be the structural and functional unit of the kidney. Let's start with part 1. So part 1 is the blood vessel that is entering into the nephron and it is wide. This is the afferent arteriole. It is wider. Part 2 is the one that's going out. So this is the efferent arteriole and this is, as compared to the afferent one, it's narrow. Okay. Now part 3. The afferent is entering inside and now it's turning into a tuft of capillaries. So part 3 is that tuft of capillaries which is your glomerulus. Then 4, the part number 4 is your Bowman's capsule. A lot of people pronounce it as the Bowman's capsule also. Okay, right. Then part number 5 you have is the first convolution. So we call it to be the proximal convoluted tubule, PCT. Right? I hope all of you know the full form. I don't have to write. Achha, theek hai, let me write also. The full form is proximal, that is close to. Theek hai? Proximal convoluted tubule. Theek hai? That's 5. Then part 6 is referring to, you see that downward dip which is there. What's that downward dip? Something is going down, it's descending. So it is the descending limb. Descending limb of whom? Part 7, the loop of Henle. Okay? And part 8 is the part that's going up. So it's the ascending limb. So basically your loop of Henle, one part of it is descending limb, then the transverse part and then the ascending limb. Okay, so 6, 7 and 8, all three of them together constitute the loop of Henle basically. Right? I guess this much is clear. Then part 9, carefully note, this is again another convolution. This part that we have, this one, again another convolution. So now it's far away from the glomerulus. So you will call this to be the distal convoluted tubule. That is the DCT. Okay? Understandable clearly. Then you have part number 10, which is the collecting duct, that finally the urine which is now formed will be collected and it will be sent out through the ureter. I guess this is clear. Okay, back to plant science questions. What's your study the setup shown and answer the following. Why do we see the reduction in the water level after some time? So initially you have the water level here. Now the water level has come down. So there's a reduction. Why so? This is because the water absorbed by the plant roots is lost by the aerial parts of the plant in the form of water vapor. So whatever water the plant root has absorbed, right, that has been lost out by the plant from the aerial plants in the form of water vapor. That is why the level of water in the test tube went down in case of A. Okay, and in case of B, what are we expected to see? There's not gonna be any change. There will be no change because the plant itself is not there. Now, what is this process called? This process, that is the evaporative loss of water. What is this called? This is your transpiration. Okay. Now, why do you have this oil used here? What's its role? The oil prevents the evaporation of water from the surface of water in the test tube. Now, normally say you didn't have this oil and you have kept this set up in sunlight for say 2-3 hours. So normally also from the surface some amount of water is going to evaporate. So little bit level of water is going to dip down, it's going to come down, right? So you don't want that to happen. To prevent that, you're putting oil so that the water doesn't evaporate from the surface. The only method or the only mode of loss of water is transpiration. Okay? The simple thing is clear. Okay, so this is again water entry. So part 1 refers to your soil particles, right? Then what is your part 2? Easy, we've already done. This is your root hair. Okay. Next, part 3 is the film of soil water around these particles, right? And 4 is your vacuoles. 5 refers to the cells from where the root hairs have come out. By now you all should be very clear. These are the epidermal cells, right? You can call this to be the epidermis. And finally, which is the final part where the water goes to and that's taken upward to the stem? The tissue is your xylem, which is responsible for the upward conduction of water and minerals to the aerial parts of the plant. I guess this is also clear. Okay, final question. Now, study the setup shown and answer the following. Name the experiment shown. This is the girdling experiment. What is done in the girdling experiment? That, see in A, your xylem is there as it is. What you have removed is, you have removed the phloem. Okay. In case of B, your phloem is there as it is. What you've done is you've removed the xylem. So now justify the observations. What is the observation first of all? In A, you are seeing that the leaves are standing out perfectly fine. In case of B, you are seeing the leaves have drooped down, right? And another observation that you are coming across is that this portion of the stem is getting swollen up. This portion has swollen up, right? Now why so? In case of A, first let us understand. So as the xylem is intact, the leaves are distended. Xylem is responsible for the upward conduction of water and minerals. We know this, right? Upward conduction of water and minerals. Okay. So that is why the water has been transported very properly. There's no problem. Upward conduction of water has been absolutely fine. So that is why the leaves have not drooped down. They are all good. No effect. Now in case of your B setup, what we've seen, xylem is not there, phloem is there. So common sense, water transport will not occur properly. So leaves will droop down. Food and food materials will be transported, right? So what we know is the leaves are the kitchen of the plant. Photosynthesis will take place. So from here the food is going to come down. And at this portion you have removed the phloem. So all the food material will be accumulated at this portion. So hence the swelling. Right? So what we are saying is that as the xylem is removed, the leaves have become drooped down. They have drooped down. Fine. No conduction of water upwards. But the phloem is as it is present. Right? Phloem is responsible for the translocation of food. Hence, there is accumulation of this food in the stem. As a result, you have this swelling. Okay, I guess this is clear. If you like this session, then please don't forget to like, comment, share, and yes, subscribe to my channel BioXplaining by Anushka. Meet you in the next video. Till then, prepare well, stay healthy. Bye-bye.